Hello there, ladies and gentlemen. This is Miss Rector. I'm going to do a little overview of Chapter 3, Hair Chapter. So, remember, I am a chemistry teacher with a chemistry background, so please excuse me if I mispronounce any of these biology terms. So, here we go. Let's talk about the study of hair. Um, there's the overview of what we're going to learn, parts of the hair, different structures, the medulla, the cortex, the cuticle. We're going to learn how they distinguish between human and non human, give some examples from the same person, how they might be different, explain how we use them in forensic investigations, and talk about a medullary index. So let's get going here. First of all, where did we start studying about this? So 1800s, you get the first sort of book that has a chapter on hair. And then it isn't until the 1900s that people really start, doing comprehensive studies of it, which you can imagine you needed microscopes to do. So that's why it isn't all that old. And then, of course, in the 30s, we've got comparison microscopes. When we talk about comparison microscopes, it's like two images side by side where you can compare them. So that is used quite a bit in forensics. And we are still finding out more now. We have neutron activation analysis as well as DNA analysis for some instances with hair and chemical tests. So even since your book has come out, things have changed even a little bit. So, your book talks about these first three phases right here, the anagen phase, the catagen phase, and the telogen phase. But now they're thinking of adding this other phase in here as distinct from this phase. So, here, this is still firmly implanted in the head. This is the exogen or the shedding phase where it's going to actually leave and then start growing again. So let's talk about all this. So, hair in mammals, what is it good for? It helps regulate your body temperature, decrease friction, protect us against sunlight. So, if you have, I have some friends with really fine hair, and they'll have to put sunscreen on their kids' heads. I've never had that problem. I have lots of hair on my head. Don't get sunburned on my head. Um, you've got the hair consists of the hair shaft, okay, and it's produced, it grows out of a follicle, which is embedded in the skin. So the hair is going to have three layers: the medulla, the cortex, and the cuticle. So if you look up here, this in the center here would be the medulla, and the cortex is sort of this layer right here where you've got a lot of the pigment. And then on the outside, that's the cuticle, kind of the scaly looking part. So the cuticle is the outermost layer. It's got like these overlapping scales, and it protects the inner part. The cortex, that's the thickest part. That's where the pigment's going to be. So if you have red hair, this will be reddish, depending on what color your hair is. And pigment commonly is denser near the cuticle, at least in humans. Now, as for the medulla, that can vary from human to human too. When hopefully we will get back in school and you'll be able to do the lab that goes with this with the microscopes, but just in case you don't, some people will have a continuous cuticle like this. Others might have interrupted cuticles, or it's sort of in segments. This might be a fragmented one, so this they look kind of regular. But here, it's just sort of randomly segmented. There could be a solid one, so pigmented area um, is filling both the medulla and the cortex. And then you may have no uh, discernible medulla, especially if you only have a small sample of the hair. Different types of hair. So here's a buckled hair, so you can see where it's sort of split and you know has a little kink in it there. The blunt hair, and here you can even see a double medulla. So the cross section could be circular, triangular, irregular, flattened. And this is all going to influence, your hair. Is your hair curly? How does it lay? The texture could be coarse or fine. And different regions of the body can vary. So your head, your eyebrows, your eyelashes, mustache and beard hair can be different. Underarm hair, overall body, like auxiliary hair, and of course pubic hair. So you might have different hair growing on your head. For example, my brother, this is not my brother, but I didn't have a picture of him. He has really blonde hair, and then when he grows a beard, it's really red. My sister has really blonde hair like this, but occasionally one thick red hair will grow in there, and just like my hair. So she's like, I have a Diane hair growing in my head, is what she'll tell me. So that's going to lead to some issues in forensics. Let's talk about the life cycle of the hair. So, again, your book talks about 3 stages. The anagen stage, this is where the hair is actively growing. Your hair grows about 1.3 centimeters per month. And the cells around the follicle are rapidly dividing and depositing that material into the hair. Then there's the catagen stage. This is where the growth is going to kind of slow down. And then the telogen stage, where the follicle becomes dormant, and this is where hairs are lost. So again, kind of adding to that, now they're sort of calling that the exogen phase, where the hair is falling out. So when you get out of the shower and there's all that hair in the hopefully hair catcher in your drain, those are these telogen phase, exogen phase hairs that are just falling out naturally. Um, a hair in the anagen phase, for it to fall out, somebody's probably going to have to pull it out. Now, notice these are being fed by the blood vessels and veins and the skin around it. So, this is going to be important because if this hair gets pulled out violently, then it might take this root with it, and that is when we can do DNA testing on the hair. A hair that just falls out like this will not have that root with it, so you won't be able to do DNA testing on that one. So, things that can affect your hair, if it's treated, bleached, dyed, all those things can affect the individual's hair. Animal hair versus human hair. So we look at the medulla is really the place to look, and the cortex, the cuticle. The pigmentation of the animal hair is usually denser toward the medulla, which in humans we said that was towards the cuticle. Unlike human hair, animal hair can change colors like bands. So if an animal has stripes, for example, they may have, orange and black on the same hair. Medullary index. So that's something we're going to talk about here in a minute. It's going to be different in animals. So if you have an animal hair, we can calculate the medullary index by taking the diameter of the medulla in the hair, dividing that by the diameter of the whole hair. So in humans, that's typically less than 0.33. So less than a third of the hair is the medulla. Whereas in animals, it's usually greater than 0.5. So more than half the hair is made of the medulla. So we can look at a chart like this and say, oh, is that cattle? Is it buffalo? Is it a sheep hair? Try to narrow it down. Also, you can look at the cuticles. So there's this type, kind of a spinous here, coronal, and imbricated here. So animals are tend to gonna kind of look like these. So here's some spinous ones and some coronal. So this may be like a dog or a cat. I kind of forget which one would look like which. I have to look at some pictures. But humans tend to look like this, sort of like, well, I kind of think of it like bricks, but their scales overlapping each other. So, microscopic investigation, you can talk about length, color, curliness. When you get to microscopic, now you can really look for some more details. Um, so, has it been dyed? If it's been dyed, how long ago was it dyed? Electron microscopes can even give us, better pictures of those scales and the pigments in them. So we can also test the hair for substances. So again, the chemicals that are in your skin are being absorbed by the hair too. That blood that's feeding it, if there's any toxins in there, it's going to go into the hair as well. So forensic scientists, we might perform chemical tests to try and figure out, were there any toxins, any drugs, any establish a timeline for any exposure to things. And then, of course, you've got neutron activation analysis, which can determine, concentrations of substances in the sample. You might also look at something. So let's say we find a body like this, and we're not sure quite when the person was killed. Well, we can take a look at the hair growth. Obviously, she's had some hair growth since the last time she was at the beauty shop getting it colored. So that might help us determine, well, let's see. She's got 1 month of hair growth here, but her last appointment was 2 months ago. Maybe that means she's been dead for a month. You can also take the segments of the hair and look for chemicals, like we said, toxins and things in them. So let's say this person was being poisoned. You could say, it wasn't here, and there was none here, and there was none here. And then for these 2 months, there was, and then there wasn't anymore. Or drug testing. If you were testing somebody for drugs, even if they haven't been using it right now, you've got these 2 months where they were. So, again, blood tests and DNA tests are things that you might be able to do if you have the cuticle of the hair. So, remember we talked about class evidence versus individualized evidence. Class evidence can only be connected to a group. So, you can say, well, I think the person had red hair, and that's as specific as you can get. Individualized evidence is something you can connect to something unique. So, DNA evidence, for example, would be individualized evidence. Only this person could fit this profile. So, again, that's only going to happen if you have the root of the hair, which you might in a violent struggle. So, in summary, we talked about different parts of the hair. One thing I forgot to mention is that you're going to find a lot of hairs at a crime scene because we're constantly losing hairs. We're shedding hairs. We have dog hair. I have dog hair on me that I've probably brought into this classroom. Even though my dog's never been in here, you're going to find some of my dog's hairs. So we're constantly transferring all this hair. We got the pieces of the hair. Hair treatments produce characteristics. Some characteristics allow you to group them. Well, we didn't really talk about the racial thing because it sort of only works sometimes, so it's not very useful. And DNA, yep, we talked about all that. Good deal. All right. You all have a great day.